Hello, welcome to Virtual Agronomy Day. My name is Pat Tranel. I'm a professor in the Department of Crop Sciences here at the University of Illinois. In today's session of Virtual Agronomy Day, what I would like to do is talk with you as I usually do at Agronomy Day about everyone's favorite weed, water hemp. Specifically, I'm going to give you an update on herbicide resistance in this species and then talk about some of the genomic approaches we're using to better understand herbicide resistance in water hemp. So for those of you that are familiar with the species, you don't need to see a photo of it again, but if you're not, this photo on the left shows you what water hemp looks like. And the graph on the right is one of the examples of the challenges that we have in trying to manage water hemp. Specifically, this graph is illustrating the increasing amount of stacking of multiple resistance that water hemp populations are doing over time. And so just to break this graph down, 1990 was the first identification of a herbicide-resistant water hemp population. It was resistant to herbicides from only one group, so herbicides that had one site of action group. Then in 1996, a population was identified that was resistant to herbicides that had two different sites of action. So specifically, it was the triazine herbicides and the ALS inhibiting herbicides. And I'm not going to go through every point, but you can see as we went over time, we had a population was identified with three-way resistance, and then four-way resistance, five-way, and then most recently a population was identified in Missouri where there's 6 different herbicide groups that one population has resistance to. And so if you have such a population in your field, you can imagine it becomes very difficult to manage that population with the use of herbicides since it already has so many different herbicide resistances within it. Before I go further on into detail, I want to step back a little bit and just do a brief review of herbicide resistance. In terms of mechanisms, there are 2 broad categories of herbicide resistance mechanisms, and those are target site mechanisms and non-target site mechanisms. Target site mechanism or target site resistance occurs when an enzyme, due to a mutation in the gene that encodes that enzyme, binds the herbicide with less affinity. So in other words, the herbicide can't bind the enzyme to do its job, therefore the plant survives the herbicide, the plant's resistant to the herbicide. This type of resistance is fairly well understood. We know the specific mutations that give rise to this type of resistance. We also can predict with reasonably good accuracy how long it will take for this type of resistance to evolve in your field, given different strategies. So if you use the same herbicide year after year versus if you rotate herbicides or if you're mixing herbicides using crop rotation, whatever, we can, with reasonable accuracy, predict how long target site resistance or how long it will take for before target site resistance occurs in your field. Non-target site resistance, on the other hand, is kind of a different beast. It's a broad category of mechanism, so it's not just one type of mechanism, but probably the most common type of non-target site mechanism is where the plant rapidly metabolizes the herbicide. In other words, it breaks it down to a non-toxic molecule in the plant. This type of resistance, this non-target site resistance, is poorly understood. We don't know the specific genes. It can be lots of different genes, and we don't know, in many cases, we don't know the specific one. We don't know what that gene is doing or what that gene product is doing necessarily. And from a management standpoint, it's very unpredictable. We don't know how long it takes for non-target site resistance to evolve. We can't predict with very good accuracy what will happen if you implement other strategies such as herbicide mixing or herbicide rotation. And so it makes it much harder to manage. Here's kind of a more specific example of the management challenge that's posed by this non-target site resistance. So with target site resistance, it's conferring herbicide or resistance only to herbicides with the same target site. Okay, so if you have target site resistance, or if you want to prevent target site resistance, you manage it by using herbicides with different target sites. Okay, so if you're anyone who's applying herbicides, you've probably become familiar now with herbicide group numbers, fairly prominently printed on the label, a number, 2, 9, 10. Those numbers refer to that herbicide target site. And the reason for using these group numbers is specifically to help you in preventing or mitigating target site resistance by making it more obvious and more easy for you to determine whether you are using herbicides with different target sites. Non-target site resistance, however, confers resistance, or it can confer resistance to herbicides with different target sites, okay? For example, metabolic resistance. If that enzyme is metabolizing the herbicide, it doesn't care what the target site of that herbicide is. It just cares whether there's a particular chemical handle or chemical group on that herbicide molecule that enzyme can attack. Therefore, we really can't tell you how to rotate herbicides or how to mix herbicides effectively for non-target site resistance, because we don't know what mechanisms are conferring resistance necessarily to which herbicides or which range of herbicides. Okay, so back to water hemp. This here is a timeline in a table format of the different types of resistances that water hemp has evolved. So water hemp's evolved resistance to herbicides spanning 7 different groups. Okay, there's more than 7 entries here because this table is further tabulated to include the specific mechanism. Okay, so don't worry, there's a lot of detail here, a lot more detail than you need to know. Not all this will be on the exam afterwards. But what I want you to focus in on is on the far right. I've indicated these resistance mechanisms by whether they're a target site mechanism or a non-target site mechanism. And you can see a clear trend, right? Prior to 2013, essentially all the resistance in water hemp was due to a target site change. This easy, predictable, I say easy, relatively easy, predictable, relatively easy to manage type of resistance. However, this has really shifted since about 2013, where all the new herbicide resistance mechanisms that have been identified in water hemp are these non-target site mechanisms, these harder to predict, harder to manage resistance mechanisms. Okay, so what's the good news? The good news, at least from a research standpoint, is we now have a high quality reference water hemp genome. Okay, so this is a paper that was published just last year. You can see there's several authors there. This is a large collaborative effort, and the title of the paper is might be a little bit misleading in terms of a genome. That the objective of this paper really was to investigate the spread of glyphosate-resistant water hemp into Canada. But in the process of doing this research, a key tool that we needed was a high-quality water hemp genome. And so that work was included in this paper. And as I said, it's now been published. So now we have access to this high-quality water hemp genome. Okay, so what? What can we do with that? Well, a reference genome enables gene identification. So in this case, genes we're interested in would be non-target site resistance genes through a genetic mapping approach. Okay, genetic mapping is definitely nothing new. Plant breeders have been using this for years, for example, to identify genes that contribute to disease resistance. And then they identify those genes through genetic mapping in the crop, and then they can incorporate that into their germplasm, okay? So genetic mapping is not new, but in order to do genetic mapping, you need a genome. And since we've just now gotten this genome in water hemp, it is new to water hemp. And it's really new to weeds. There's really not very many weed genomes that have been made available yet. So just real quickly, genetic mapping, the way it works is you cross a resistant, in this case would be a herbicide resistant plant with a sensitive plant. And what I've illustrated here are the chromosomes, or the pair of chromosomes. This is a simplified example showing just one pair of chromosomes. So you cross that, you generate an F1, which would get one of this pair of chromosomes. One would come from its mother, one would come from its father. You take that F1, you cross it with itself or to another F1, and then you would get F2 population. And what happens is you actually get recombination between these pairs of chromosomes. And so this is illustrated in those F2s there, where you have a mixture of, in this case, the orange or light blue, a mixture of genes that are coming either from its resistant parent or the sensitive parent. So what you do then is, since you have the reference genome, you can determine for each of these F2 individuals at which part, or at every location in the genome, whether that genome region came from the resistant parent or the sensitive parent. And then you score them for the phenotype, the trait of interest, in this case, whether it's resistant or not. And you look for regions of the genome that are shared by the resistant individuals, but not possessed by the sensitive individuals. And so in this hypothetical example, you can see that this part of the chromosome that I've put the box around, that region would zero you in on probably the region of the genome that has the specific gene that you're interested in. So this is cool. One of the nice things about this is that you don't have to really go into this having any idea what that gene might do, okay? This simply will get you to the region of the genome, whether that gene is involved in metabolizing the herbicide, or maybe that gene turns on another gene. You really don't need to know that. You can go in there, search through the genome, and it will lead you to that gene there that's responsible. So we're currently actually using genetic mapping to identify herbicide resistant genes in water hemp. In terms of 2,4-D resistance, we've actually identified a relatively small region of the genome. that includes a particularly cytochrome P450 gene that potentially could be involved. And so we're now narrowing down that region of the genome to see if that particular gene really is responsible. We're also using genetic mapping to identify genes involved in HPPD resistance. This is proving to be more challenging. It looks like resistance to HPPD inhibitors is actually conferred by multiple genes, or several genes may be involved, and so you have to do the genetic mapping sort of for these multiple genes that could be involved. So, what does this mean? Why do you, as a farmer or weed management professional, why do you care? So I'm going back to this graph where we're showing increasing cases of multiple resistance. One of the questions that we really don't have a good answer to is when we have this, multiple resistance where you have, let's say, 6-way resistance in a population, does that mean you have 6 different mechanisms and each of those mechanisms is conferring resistance to a particular group? Or might you have one mechanism that can confer, maybe not resistance to all 6 of those herbicide groups, but maybe you have one mechanism that confers resistance to 2 or maybe 3 of those herbicide groups. So maybe you have an enzyme that can metabolize an HPPD inhibiting herbicide, and it also can metabolize a chlorosetamide-like herbicide. And so that one enzyme is giving resistance to 2 different herbicides, or herbicides from 2 different groups. So that's, without having the answer to that question, we really can't tell you and predict with good accuracy of what different herbicide resistance mitigation strategies will have. If in fact one enzyme can metabolize both a chlorosetamide and an HPPD inhibitor, telling you to mix a chlorosetamide herbicide and an HPPD inhibitor herbicide into your weed management strategy is not going to be very effective from a resistance management strategy. So, in summary, herbicide resistance in water hemp continues to get worse, and particularly we're concerned about the increasing cases of non-target site resistance in this species. Genomic approaches will give us a new avenue to better understand these non-target site resistance mechanisms. So this is an exciting time as a weed scientist, and hopefully this information will ultimately lead to better management strategies for you, the growers. However, I do want to conclude by saying that, regardless of how well we understand herbicide resistance, as we're stacking more and more cases of herbicide resistance into water hemp, and we have a finite number of herbicide options out there, increasingly we need to think about and incorporate non-chemical strategies into our water hemp and our weed management strategies. So with that, I wish we could have been here in person. I hope that we can be in person next year at Agronomy Day, but I do appreciate you tuning in and listening to our virtual Agronomy Day. I wish you safety and health, and go Illini!